Hello, everybody. I'm Dr. Lara Strittmatter, and I'm here to summarize the findings in the poster entitled Method Optimization for Quantification of Retroviral Particles in Unprocessed Bulk Material by Thin Section TEM. In this poster, I'm going to summarize quickly at the beginning the technique involved in how to do this testing, and then I will focus on the different aspects that we did for optimizing it. So the technique consists in starting with clarified UPV material and doing a centrifugation to concentrate the viral particles, and then on that concentrated pellet, process it for TEM, and then visualizing the particles. Since it's not a direct count technique, what we are doing is we are sampling an area of that pellet, and in that counting the particles and then extrapolating that number to the full volume in which the particles were found. And in that, we are calculating a titer. The particles that we are looking for are the ones that you can see in the poster illustrated in the circles. Those are C-type retroviral-like particles. They are produced by cell lines that harbor endogenous retroviruses. And when these cell lines are used in the production of biologicals, these particles get released into the media and remain with the product. So the whole purpose of the test is to calculate how many of them are present, so the manufacturer can then calculate the reduction log necessary to eliminate them from the product. So with this, this technique is not a new one, but because we were bringing transmission electron microscopy and this technique to Eurofins, we had an opportunity to update and revise this method and incorporate new updates and resolve some of the challenges that this technique presents. In the poster, I'm going to be talking about those two. In one half, in the left-hand side, you have the challenges that the sample preparation itself presents, and they are described in the table 1, and then I'm presenting some of the solutions. And then on the other half, on the right-hand side, you have the updates that we did to the imaging technique and the sampling strategy, also to try and make the test more accurate and more repeatable. So one of the main challenges that we found is that because the new biologicals making has improved so much, now we have a much better yield of product, and that protein remains in solution together with the viral particles. So in that first step of centrifugation, when we are concentrating the particles, the protein is coming down too. And that makes a bigger pellet, a more compacted pellet, and then it also interferes with the homogenization of the particles, which restricts that evenly distribution that will help in the sampling. So what we tried were 2 different centrifugation schemes and using different volumes of initial material to see if we could mitigate this problem. So we tried a neat concentration and then half that volume, and then we tried 2 different centrifugation schemes, a high speed, short time, and a low speed for a longer time. And the one that worked the best, as you can see in the pellet homogenization section of the poster, was the half long, which is having less protein in the media and compacting it less, allow for a better distribution of the particles and a much more consistent sampling that was reflected in the lower coefficient of variance that we found when we analyzed these samples. And then on the sampling methodology, I just want to point your attention to, we started using an automated imaging method to try to get operator bias out of the mix. So we select the areas at a very low magnification where we cannot see if there are particles present, but we choose the areas based on the integrity of the section itself and if there are no stains or blemishes on them. And then the computer on its own takes the pictures, and then we compose a montage that is made of approximately 100 high-resolution images, and that's where we count the particles. So in all the poster, you're going to see little yellow dots. That's how we count the particles. And even though you cannot see the particles present, the way they look is like in the pictures. So wrapping up, we found that for high protein content samples, the best solution is to try lower volume and low speed centrifugation scheme.